Good afternoon and good morning, everyone. My name is Renee Boucher, Events Coordinator, and on behalf of Simulations Plus, I welcome you to today's webinar, which will focus on the exciting new release of the standalone PKPlus software, the next generation tool for preclinical and clinical trial data analysis. Walt Woltosz, Chairman and CEO of Simulations Plus, will be presenting today. Joining Walt as panelists are Mike Bolger, Chief Scientist, Vera Lukachova, Team Leader, Simulation Technologies, Denise Morris, Assistant Director, Clinical Pharmacology, and Inger Darling, Associate Director, Clinical Pharmacology. An opportunity to ask questions will follow Walt's presentation today. You may either send your questions using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you're using a telephone to listen to the call, please be sure to enter the unique audio PIN displayed when you join the call. When you enter your audio PIN, this enables us to unmute your line so that you can be heard. This webinar is being recorded for playback at our website, www.simulations-plus.com. Also, upon request, the presentation slides can be made available and shared with colleagues. It's now my pleasure to turn the presentation over to Walt Woltosz, Chairman and CEO of Simulations Plus.
Thank you, Renee, and welcome everyone to our PKPlus webinar. I know many of you have been patiently waiting for far more months than we had expected, but we finally achieved it, and PKPlus has been released. I'm going to try to give you a quick overview with a few slides and then go right into a live demonstration of the program. But first, just wanted to summarize the features of PKPlus. It is a Microsoft Windows program. We designed it to provide all of the assistance with 21 CFR Part 11 compliance that we could. So Audit Trail and validation are an integral part of what we've been developing. One of the major features, and one of the reasons we call it next generation, is point-and-click ease of use. There's very little typing involved. It's rare that you would need to type anything. We can do a complete clinical trial data analysis without hardly ever touching the keyboard. And that minimizes errors, typing errors, and also makes things go very quickly and maximizes productivity. We do import common data file formats, the comma separated variable and SAS XPT files. The file mapping, you'll see, is very easy to do to the internal display names that we use inside of PKPlus. We base the program on a relational database, and that allows us to do some really interesting and powerful things for data preview and exploratory data analysis, as well as setting up analysis data sets with specific filters and VLQs and exclusions. Non-compartmental analysis, very easy. Compartmental analysis, 1, 2, and 3 compartment, or any combination of those, very easy to do. Report quality output in docx format and in Excel format, both automated and custom reports. And then a value-based licensing approach. We do not require a named user license, for example. You can share a license among your colleagues within the same geographic area. For 21 CFR Part 11, there's a website here at the bottom where we borrowed this quote. It's not possible for any vendor to offer a turnkey Part 11 compliance system. Any vendor who makes such a claim is incorrect. Part 11 requires both procedural controls, that is, notification, training, SOPs, and administration, and administrative controls to be put in place by the user, in addition to the technical controls that the vendor can offer. At best, the vendor can offer an application containing the required technical elements of a compliance system. And of course, audit trail and validation are key elements, along with preservation of input data and model settings. And we've provided that in this next generation PKPlus software. For the audit trail, for now, we're requiring only a username. We may require a password at some time in the future, but just having a username and your own procedural controls within your organization, we felt was a little bit more friendly. You don't have to worry about remembering a password. If you're worried about your users sharing the same username, then maybe you want to have a little talk with them so they don't do that. Or if there's really a demand for a password, we'll add that in a future version. All source data is saved within the project database, so once you load your source file, you never go back to that file. It's saved and preserved in the database, so even if the original file is changed or lost, the data is preserved. So years later, you can go back and it's all there in a single package in the project database. There is an activity log that tracks major actions. So, for example, who did what and when. So all the major actions are tracked. We don't track every key click or mouse click or keyboard action. We feel that's excessive. What you need to know is who ran what, when did they run it, what were the settings, and so on. Once an analysis is run on an analysis data set, that data set is locked unless you delete the analysis. So let's say you set some filters, some BLQs, some exclusions to a particular data set, and you run an NCA, and then you say, well, you know, I'm going to change some of the terminal slope points. You can't do that because that analysis has that data set locked. Now you can copy that data set in a few seconds, make a second data set, and change it, and then change your terminal points, BLQs, exclusions, filters, whatever you like. But each analysis that has not been deleted preserves its data set so that you have a complete audit trail. And analyses can be deleted as long as they have not been archived. Once you archive a data set, that says, I am done with this. I'm going to use it in a report, perhaps to a regulatory agency or back to our sponsor company if you're doing this as a consulting project. And once it's archived, only your PKPlus administrator appointed by your organization can delete that analysis. For validation, we've validated models by comparing NCA and CA results, CA being compartmental analysis results, using all possible model settings and running on a variety of actual and simulated analysis data sets. We compared these with other methods, for example, R and other Excel models, for example, with no significant differences in numerical results. You'll always get a little machine noise difference here and there, but nothing significant. And we provide the validation data sets with PKPlus with the results you should achieve. It does require manually running these validation data sets. It's not automated yet. In a future release, we hope to offer batch mode testing as an option. But the program runs very quickly. You can run the entire validation data sets and compare the results before lunch. We began the design of PKPlus using the FUSE, or PHUSE document that was published in 2014. So the automated reports that the program generates are based on this particular document. But we found that in talking with many people who do non-compartmental, and this document applies only to non-compartmental analysis, that people don't really adhere to this strictly. It's a general guideline. And so we've enhanced some of the things a little bit from what the FUSE document provides. Finally, the licensing. All of our software products use the same flexible licensing. We use the Flexera license system. You can install on a standalone or on a server. If you're on a server, you can share them in the local geographical area. This is typically, someplace that you can drive between sites within an hour or so. If you're on the East Coast or the West Coast, then the license policy requires separate licenses. Standalone licenses can be shared, so we don't have a named user requirement, and we have affordable pricing. The first annual license is under $4,000. Again, it's shareable, and discounts are provided for multiple licenses at different levels. So that's been making people very happy. All right, and now I'm going to get out of PowerPoint and go over to the cloud, where I have already started PKPlus. And you can see it's asking me to enter my username, so I'm going to enter my name. Hit enter, hit a return key. And I'm going to start from scratch here. I'm going to move this over a little bit and click on Create Project. You can see there's a bunch of projects that are in the cloud already from various tests that we've been running. And I'm going to call this Demo for Webinar, if I could spell right. Yeah, it still blew it, didn't it? Demo for Webinar. And I'm just going to use some test data. So, okay. It creates a project. PKPlus is organized by projects. And within a project, we have some files that will be saved for you that relate to different things. But almost everything is saved in the database itself. The database is Microsoft SQL, or System Query Language, SQL, we also say, SQL Server Express. When I load the source file, I can load either by saying File. I've already created the new project, so File, Open Project, or Open a Source File. I can do it that way, or I can come down here to Source File and right-click and say Open Source File. Either way. And I've got a little data folder here, and I'm going to load this particular test data set, test1 CSV, double-clicking on that. It's now loading that CSV file, and now this file is in the project. I will never have to go back to that file, although I preserve the name of the file and the path to it as part of the project. And you can see the raw data in this file. The raw data file consists of a row of headers and then rows of data. Each column is a particular variable. So here we have study arm. In this case, study arm in this data set was designated as oral or IV. Different series, if I scroll down, you'll see series 1, series 2. So there's 2 different series that we're involved. The subject ID, of course, there's various ways of representing subject ID, and we're going to map that to the internal name that we use in PKPlus. Time, concentration, concentration units. Dose time in this file was actually a date. You can see here's April 5th, 2015, March 5th, 2015. The dose amount, the formulation, tablet or IV, is in the raw data. The treatment, in this case treatment, was used to designate fasted or fed. There's some fed. The age of the subject, the age units. This column, X dose, you can see basically duplicates the dose but has the milligram designation on there. Sex, male or female, and body weight in kilograms. And so this is the raw data or source data. I'm going to map that data by going to the next tab. And the flow in PKPlus is left to right, top to bottom. So very intuitive flow in the part of the world where we do things left to right first. And so here I'm going to go study. I'll click on the drop down and I'm going to map that to STDY. Arm. This was our oral or IV. In this case, study arm. Series number. Fed fast. Well, that was called treatment, TRTP. Subject was sub-ID. Sex goes down here. Sex. Age. Age. And age units. It already defaults to years, but I'll go ahead and map it anyway. Age year. Weight. And it's in kilograms. Dose. And default units, milligrams is correct. Route. Well, in this case, the route and study arm are redundant, but I'll just map it that way. And it does want you to have a route. Time. And concentration. And concentration units. We also had some other variables that are not part of the default list here. We had formulation, for example, and we had the dose time, which was a date. So let's map those two. I'll add a variable. Go to formulation, F-O-R-M-N. Say OK, and map that to formulation. And then I'll add another variable, dose date, which is called dose time in the raw data. Okay, so double checking, everything looks okay. I'll apply the map, and it says, all right, what is the internal designation for IV? Well, in this case, these are bolus dose, so IV BOL. And what is the internal designation for oral? Well, extravascular. Set those two, say OK. The map is applied, and it will automatically move forward to the mapped data tab. And we can see now the mapped data under the PKPlus display names. Double check it. Everything looks fine. We'll say save the data to the database. Click here. If I needed to reset the database for some reason, I can come back to this tab and click on Reset Database. So if I found out later that I made a mistake or I left something out, I could do that. Automatically, they program advances to the Data Preview tab. Now we can look at any subject. We can see for subject 26, we have 3 profiles. Subject 27 and the rest, we have 6 profiles. I can look at any combination of these to see what they are. But I think one of the most magical parts of PKPlus is this feature, groups. PKPlus examined our map data and automatically created all of these groups. So anytime it can recognize a grouping within a set of map data, or within a column, it will automatically create those groups. So automatically we've got female and male, we've got the different dose levels, we've got fasted and fed, we've got oral and IV, and so on, different dose dates, different studies, arms, and series. And this is quite powerful. So if I want to, for example, look at all the female and male together and compare those 2 groups, I just hold the mouse button down, hover over and select those, and click here on View Selected Groups. This will bring up the plotting window, and it will generate profiles and statistics for these 2. There's about 150 profiles here. If I click here on log concentration, now if we turn off the female group and turn it back on, you can see there's a pretty good overlap between the 2. So we're not seeing a strong difference between those 2. Now the scale is changing when I do this one. So if I go back where they're all on and say auto fit off and then change, you can see there's quite a few more males than there are females in this particular data set. But this, you can see the value of being able to very quickly look at sex, at fasted versus fed, for example. Do we have a significant food effect? And here, of course, we're seeing both IV and oral doses at the same time. So here, if I turn off the fed group, you can see it's pretty well the same distribution. Well, that's interesting, but really we didn't have any fed in the for the IV doses. So what we'd really like to see is, let's say, just the oral doses. So we can add a new group. And I'm going to say oral. In fact, I'm going to do something even fancier. I'm going to say I just want to see the female, keyboard here after that, female, oral, fed, 140 milligram. So I created this new group here, female, oral, fed, 140 milligram. And to create that group, I'm going to add filters. So first, I'm going to select sex equals female, apply that, sex equals female, and route equals oral, apply that, and fed fast equals fed, and dose equals 140. Notice I have not typed anything. Everything's point and click. Now if I look at that group, it turns out there's only 2 that have this particular dose in the fed state. And I am seeing a significant difference between these 2. So I wonder why that is. Well, this is subject 100, 27-year-old, 64 kilogram, 140 milligram oral fed. Dose date was May 5th. And the other one, 48 years old, difference in age. Not much difference in, I'm sorry, I'm subject 48, age 25, age 27. So no significant difference in age or body weight, but a significant difference in the concentration time curves. So I might ask myself why and do a little bit more homework if I have another source of data on those 2 subjects. But you can see the value of having the relational database. It automatically allows us to create all of these different groups for you, so you can very quickly do some exploratory data analysis in a matter of minutes. You can quickly see if there's something that jumps out at you as a difference in sex, a difference in fasted or fed state, is there linearity in the doses. So that's a very powerful feature, and again, the ability to create your own groups with multiple rules is a very nice feature. Now, if I want to do an analysis, we would go to the next tab, Analysis, and we can create analysis data sets in the same way that the groups were created, or we can push any of the groups forward to be an analysis data set. I'm going to click on All here, so this is everybody, and view the selected groups. Still in the Data Preview tab. And now you look at the plot, and what jumps out at you right away, excuse me, is we've got, it looks like 2 profiles here where we've got a missing value. So we don't want to use that. I'm going to right-click on that point and exclude the point. And it wants a reason. I'll call it a 0 value outlier and exclude that. Now we see this is excluded and the comment that goes with it. We've got another one here. Exclude that one. You can see that this one is in profile 129, by the way, the one I just set. This new one is also a 0 value outlier. Exclude that one. And now we've got a cleaned up data set. If I scroll down through the data summary here, and you can see we have automatically all of the concentration time points for all of the profiles highlighted. If I wanted to look at just a certain profile, I can click on it, or a certain group of profiles. So very, very quickly, the program can allow you to look at any combination like that. To go back and highlight them all, I can get the scroll to go all the way down here. I'm holding the Shift key and the down arrow to do this. And we've got 150 going off the screen a little bit here, but there's 150 profiles. If I scroll down, I'm going to scroll down using this, and you'll see that there are some time points here where there's no observation. This is a 5-minute observation. We only have that for the IV doses. So it's pretty easy to spot in this particular data set the IV doses, because the IV doses have an observation at 5 minutes where the oral doses do not. You can see in the data tables, you can look at any of the profiles. Okay, here, here's a profile. We've got something that doesn't have a number. It's got BQL. So I could exclude that using the exclude feature, but instead I want to show you the BLQ feature. So I'm going to exit out of this plot window, and for this group, all, I'm going to go to BLQs, add a BLQ, and when alphanumeric it says BQL, we use BLQ in the program, but sometimes people use BQL. If I see that, I'm going to treat it as missing, and throughout the full profile. So you can set a BLQ based on a numeric value, and then the action based on the trigger. Do you want to impute a certain value, treat it as missing, or go ahead and use the actual value? You can type in a reason if you want, and then where do you want this BLQ to apply, before Tmax, after Tmax, or the full profile. So it keeps track of that. I don't have any filters because this is all of the profiles. I haven't filtered them. And you notice the profile number, that's something assigned inside of PKPlus that basically condenses all of this information that makes this profile unique into a single number. And this can be handy for reporting where you don't have to show all of this in a table. You can have a profile cross-reference table below your main table without cluttering it up with a lot of information that can be summarized a little bit more briefly. All right, well, I want to copy this to the analysis data set, and I'm going to call it all with BLQ. I have to watch my time here. It's easy for the time to fly by. Now I'll go to the analysis tab. I have all with BLQ. I can view that data set, and it will look just like it did on the data preview under groups. 150 profiles, doesn't take too long to load. So here I have this, and I think it was profile 3, wasn't it? Yeah, so there's the one that did have BQL, and now it says we've called it BLQ and treated as missing. Now to run an analysis, I can choose for non-compartmental analysis, and by the way, I can modify filters, BLQs, and exclusions on this data set until I run an analysis. Remember, once I run an analysis, this data set will be locked unless I delete the analysis. And that would be done over here on the Results tab. So every time I run an analysis, it will have an analysis ID. The model settings will be shown over here. We'll come back to that shortly. But as long as that analysis is still here and I haven't deleted it, then it will lock that data set so that I can't go back, change terminal slope points or BLQs or filters or exclusions, and end up not having the audit trail for that analysis. So if I try to run this analysis without setting the terminal or double checking the terminal point slopes, click on Run NCA, it's going to give me a message. Please confirm the terminal slope points for this data set. So it wants me to do that. And now I'm going to click on View or Edit the terminal slope points. When I do, a plot window will show up, terminal point selection, and you can see all of the profiles here. You can see the terminal point slopes that were selected by the algorithm. It follows the fused documents, so we use the maximum number of data points that gives us the highest adjusted R squared and more than 1.5 times the half-life if possible. Sometimes it's not, and that's why we want to double check. So I'm going to go down. I'm just going to hit the down arrow and step through these. You'll notice up here this little window gives us the slope and intercept R squared, adjusted R squared, and this status, success. That means it survived all of the rules. We've got at least 3 data points. We've got more than 1.5 times the half-life, and so on. And so as long as we've met all the rules in the fuse document, then we call it a success. So I can just tap the down arrow, and in a matter of just a few seconds, I can check all 150, and my eye is focusing, sorry, my eye is focusing on the word success here. So as long as I'm seeing success, I'm just tapping on the down arrow key to step through all of the different profiles. And you can see in just a matter of seconds, we're going to go through 150 different profiles. And you can see on the table on the right how many data points have been included in the terminal points as well as on the plot. You can also see the profile number up there just to the upper right of the box that says success. And almost to 150, 150. So they were all successful. Now that I've done that, now I can run an NCA. But actually, before clicking that, we've got all of the 4 options that are typically used. So linear, linear end log, linear log, and linear up log down. I'm going to choose linear up log down. This is single dose data set. It's not a multi-dose data set. Partial AUC, if I say yes, I want to do a partial AUC or more than one, that's my begin time. Or maybe I want to go, say, from 5 hours to 10 hours, which are not observed data points. And it's going to have to interpolate. So I could add more of these, but I'll just do that for now. Again, I've got to watch my time. Run the NCA. Profile is invalid for data analysis. Profile 78, time 24 hours, concentration as one of these. So I didn't catch one of those. I'm going to stop that. Go here. Go to view the selected data set. Go to profile 124. 124? Did I remember the wrong number? Somebody help me.
It was 78.
78, thank you. I don't know why I got 124 in my head. 78, so right there, profile 78. All right, well, one way to do that is to just use this pound value exclamation point as an exclusion, or to go to I still want to go to 124. 78. Okay, 78. Right click here, exclude the point, and I'll say again 0 value outlier. So now we've excluded that point. We'll go back, run NCA, and it says check the terminal point slopes again because you might have changed something. Well, all you really have to do is open that window. I'll go to 78. Success, still good. Close the window, and I'm good to go. Run NCA. This status window, analysis complete, so it's run the 150 different NCA calculations. You can look on our log plot. We can see all the terminal points that were used here. Pretty busy, but again, you can select individual or any combination of profiles, your choice. And whatever profiles you select, you'll see the NCA results across here, and you'll see the statistics on those down here. So arithmetic mean, standard deviation, CV, geometric mean, median, min, max, min and max. Excuse me. So a very fast process. We've run the NCA now on everything with the BLQ. If I go to the Results tab, you'll see now Analysis ID 2. It skipped the number 1 because the number 1 was incremented when I tried to do the first NCA, but I still had that pound sign value in profile 78. You can see this analysis was a non-compartmental analysis, the name of the data set, the model settings that were used. These are all preserved in the database. View selected result, that'll bring up the same plot window that we had earlier. And then we see that there are some tables, figures, and lists. We can select them all. And I'm going to tell it to generate all of these. I'm going to ask it to open the file location when finished. And I'm going to click on Auto Reports. And I select where I want the reports to go. I'm going to go to a folder that I created earlier called Results on the desktop. And my window is a little taller than what my display is allowing us to see. So I'm just going to shorten that window. Select folder. This is disabled right now as it's generating the reports. And when it finishes generating reports, it will automatically open the results folder so that we can look at those files. So it's generating a series of docx and Excel files. Oh, did I forget to change the path? I do that all the time. File reports, desktop, results, cancel. It should be in there. Well, why isn't it opening that window, that folder? Is it behind something? No. All right, let me do that again. I'm not sure what I did. The data set is selected. I selected the reports. Auto reports. Results. Click here. What's going on with the cloud? It won't let me open that folder. It's not hidden behind something. That's weird. It always works. It always works. Can I open any other folder? It looks like we've got a cloud issue here. Let me try a custom report. The custom report generator lets us choose any combination of profile variables, which includes the time and concentration, and the analysis results, which includes, in this case, because it was a non-compartmental analysis, all of the possible calculations for analysis results. So in this case, let me just try and see if I can get this thing to cooperate. Study, arm, series, subject, I'll go time, concentration, and those, and put those in the report. And then for analysis results, AUC to infinity, AUC to the last observation, extrapolated AUC, number of points, and EZF and CLF. Put those in the report. We could filter, so even now that we've done the analysis on all the subjects, we could still filter by female, oral, 140 milligram, fed state, or any combination of filters that we want. So it's your choice. When you're running an analysis, you can run it on a specific data set where you've already filtered down, or you can run it on everything and then filter down here at the report stage, your choice. For tables, this is a test table, I'll just call it. And for the title, I'm going to say 24-point bold. For the header that's just under that, I'm going to make that, say, 14-point bold. And for the column headers, I'm going to go to 12-point and bold. And for plot options, I'm going to say I want both individual plots and an all-in-one plot, and I want to see both linear and semi-log. And I can put the titles on here and so on. For the path, I'm going to go again, try again, for the results folder. Select folder. And I'm going to call this webinar report all. And generate report. And it's done. Let's see if we can open this now.
Walt, can you try to scroll to the bottom of the window? I think you have that whole, and there are some open folders at the bottom. If you click the folder at the bottom of the taskbar.
Oh, cool.
Yeah.
It's still not showing up, though.
Try to right-click, close, and reopen them at the bottom of the taskbar. For some reason, they must be pushed off the screen.
Did I close them both? I guess I did.
Yeah.
That's very strange. I've never had this happen before.
Oh, they are open somewhere.
Yeah, well, where are they?
Can you open any of the others just so that it opens any window so that you can navigate?
Yeah, I tried that earlier. See, it won't open any window.
Minimize your PKPlus and see if they're behind it.
Yeah, I tried that. See, not there.
If you go again to the bottom of the window, if there is a scroll bar to the right, maybe the window is just too wide. Oh no, there isn't. Oh, there they are.
Oh, thank you, thank you. Let's see if I can move these over. Yeah, I can see them. Well, that's interesting. I've never had it do that before. Huh. Okay, so there's all of our automatic reports that we generated. Let me close this extra window here. And so we'll look at first the automatic reports, individual PK plots. And we've got a unlicensed version of Word on here that keeps popping up that little thing. We'll have to put a licensed version. But here's on the automatic report, so individual PK plots, you see the data set that was used, the run date and time. This is based on universal time. You have the username and the project name. Linear view, log view, the specific data set info that makes this profile unique, and the times and concentrations. I don't know what this window is doing, but it looks a little funny. All right, so this would scroll down to all of the other individual PK plots with their times and concentrations and specific data. So that's one of the automatic reports. What did I do? Don't tell me I clicked on the wrong close. All right, there we are. We're back again. So now I want to close this Word window here. And now list of individual PK concentrations and times. One of the other automatic reports. So that. So we've got our same header information. We've got all of the mapped variables and the concentrations and times reported in table form. Individual PK parameters. So here we have a little bit of wrapping because we've got so many columns here, but you can see all of the NCA results for each of the 150 profiles. Mean PK plots. Okay, so this just gives us our whisker plots, the mean and spread for each of the linear and log, semi-log views for all of the 150 profiles. Summary table of PK concentrations. So this gives us for each time point the number of points and then all the appropriate statistics for each of those time points across all of the profiles that were used in this analysis. Summary table of PK parameters. And so here's NCA results and the statistics for those. And then we have an Excel file which gives us 3 different tabs. So you've got the individual PK concentrations, individual PK parameters, so the results of the NCA, and the summary table of PK concentrations with their statistics in Excel format. So between the docx and the Excel formats, we have that set of automatic reports. So that's the auto reports. And then the custom reports, in this case for demonstration, I selected a few parameters that were profile related and a few that were analysis related. And you see here, for this particular profile, we don't repeat all of these columns because they would be redundant. It's a lot easier to read the table if we do it this way. So you see for each profile, the mapped variables for that profile and then the concentrations and times. Okay. Now I didn't select on the table tab, I did not select statistics, but you have a selection of a complete set of statistics or any subset that you can use. Let me show you that. Custom reports, table options. Over here, I can select all of these statistics, or any subset of those, and those would be provided below the tables that I just showed where we had the individual concentrations and times. Okay. This is the Excel file. So once again, it gives us the individual concentrations and times and mapped variables for each one. And then finally the plots in docx format. So we ask for linear and log. And then we see all of the data that apply to each set of plots, times and concentrations, and map variables that make that profile unique. And if I go all the way down to the bottom, then I also ask to see all of them together. And here you can see the color coding that's used to try to demonstrate or separate the different ones. And then this was profile 150, so this is the last of the individual plots. Okay. Well, sorry for the confusion with the results folder there. Thank you, Viera, for helping me find those off to the side. So we've got about 12 minutes left. There are a few more things I could show you, but I think it'd be more important now to open the webinar up for questions and see if we can answer any questions that you might have after seeing what we've been able to show you in about the last 40 minutes or so. So Renee, are you going to manage the questions?
Sure. Xiaohua Gong has a question asking, what if the data set contains a mixture of single dose and steady state dosing? That is, there is only a single dose recorded at steady state. And what about a mixture of single dose and repeat doses? That is, there may be multiple doses recorded per subject.
Right. Well, presumably there would be something, some identifier in your raw data that would allow you to distinguish between the single dose data sets and the repeated dose data sets. And so if that identifier is, for example, a series number or some other column, it doesn't have to be one of the existing default column names. You can have a column with any name you want. Then the program would automatically create a group for that, create two groups for that, single dose and multi-dose, or you could create the group if it didn't automatically create it using some other combination of mapped variables. So you'd have separate groups for the multi-dose and the single dose. That's what I would do.
Okay, and Kathy Lester asks, has PKPlus been validated against Win non-Lin?
We have validated against a variety of programs, including a very popular program with that name.
Okay, Sue Jun has a couple of questions. What type of files can be imported is the first.
CSV and SAS XPT files. The file format currently must begin with a row of header variables, like the first row you see here, and then followed by rows of observations and other variables that go with those. In a future version, we'll allow comment lines to appear above this first row of headers, but in version 1.0, you must begin with the row of headers.
Okay, and the other question from Sue is, are CDISC standards covered for import-export?
I do not believe they are. Does anyone else have a comment on that from the panel?
From an import standpoint, you can import your CDISC data set, so you can import an XPT. I'm not sure, Walt, on the export side. I think that that's something that may be considered at a later date, but definitely for import, you can import your standard CDISC data sets.
Thank you, Denise, or Inger. I'm not sure who that was.
That was Denise.
Oh, thank you. Thank you, Denise.
Okay, Sukesh Vodoganti has a couple of questions. What if a data set show are not success?
I'm sorry, I didn't understand that.
The question, it's probably my ignorance, what if a data set show are not success?
I'm guessing that I'm guessing that question refers to the NCA terminal slope where you were looking at it, if terminal slope was successful. When you were reviewing the terminal points, right?
Yeah,
you were looking at the
That's
points.
exactly right, yeah.
Okay, so here. So if we had one that was not successful, let me just, data set is locked. See, I tried to change it and it won't let me. But I can copy it, copy to a new data set.
You might also demonstrate an answer to another question about whether you can choose data points.
Yeah. So this one is not locked because it's a new data set. And now if I were to right-click and say remove this from the terminal slope, I still have success. But what if I remove this one also? All right, now the status is less than 1.5 times half-life and less than 3 points. So it's telling me, that's violating the rules. I can go ahead and use it if I choose to, but I might want to just go in and, let's say this data point was way down here or way up here. It was clearly an erroneous measurement. I might just say, well, skip that data point, add this one to the terminal slope instead, and so I can manually choose any of the data points. If I want to keep adding data points, I can do that, and I can see what the adjusted R squared and other numbers are here. So you have complete control over the terminal slope points. The other thing you can do is, instead of automatic selection with the opportunity to override it, you can say, don't use any terminal slope, and then you won't get an extrapolated AUC. It'll just go to AUC last. Or you can say, I want to use a fixed number of points. I always want to use, say, 3 points, or I always want to use 4 points, or some number. So you have control over how the terminal slope points are selected. In general, what I find is let the automatic method choose them and then inspect them and change them if you like. If you say, all right, I really don't want that point on there, you just go up there and remove it from the terminal slope. And you'll see which ones have been selected, which ones have been skipped. In this case, we skipped the 12-hour point, and the calculated values using the selected points. I think that answers the question.
Okay, we have a few questions from Pooja Manchandani. Other than NCA, can we do covariate analysis as well as the first?
No, we do not do covariate analysis. One thing I didn't demonstrate was the compartmental analysis. Let me just choose a very small data set. This one only had 2. Copy that. Email, oral, fed, 140 milligram. Okay, I'm going to use that just to save time because we are running out of time. And I'm going to say I want to run 1 and 2 compartments. You can see any combination here. I want to fit each profile. In this case, there's only 2 profiles. You can see here, 2 profiles. Default weighting is 1 over y hat squared. The route is mapped, the dose is mapped, the body weight is mapped. Run the compartmental analysis. And you can see here now the results. So I have the 1 compartment. I can turn those off. 2 compartment. Look at it on a log plot. It's nicer to see typically. In this case, 1 and 2 compartment not so different from each other. But in many cases, of course, the 1 compartment is inadequate and you need a higher order model. And then you see your fitted parameters here and the statistics on them down here. It does take a little bit longer to run compartmental. If we ran it on the full 150, and we don't have time for that, that would probably take 2 to 3 minutes, I would guess, to calculate all of those for both models fitting every single profile.
Okay, Anand Ketkar asks if can bioequivalent analysis be performed?
Not at this time in the traditional sense of bioequivalence. The IVIVC module in GastroPlus is what we would use for that.
Okay. Is PKPlus able to do composite data set analysis?
I don't think I know what that means. What, Denise, Inger, somebody, you know what that refers to?
Yeah, the only thing I
I don't know if that refers to doing IV and oral at the same time or things like that, which it can do.
Maybe we could get a clarification on the question if that was not the answer that they were looking for.
Okay, we have a couple of more questions. Can we analyze the patient profile if the dose and infusion rate administered were different?
Can we analyze it if the dose and infusion rate were different?
If they were different for each patient, that was the follow-up question.
I don't see why not. Does anyone see any reason we wouldn't be able to? I mean, it would be in the data.
Yes, so yeah, I agree. You can do it, you can map it, right? So if in your input file, if you have information about the infusion rate and time for each patient as they differed, you can map it in this so that the program knows how they differed for each patient, and you can do that analysis.
Yeah, I mean, the NCA and the compartmental analysis, NCA is always run on each profile by itself. Compartmental analysis, you can fit each profile or you can fit across profiles. It's your choice. So if you're doing something where there's significant differences among profiles, I would fit each profile separately. If everything's the same and you want to fit the best average model across all profiles, then you can fit across profiles.
We have another question. In case of multiple administration, are specific PK parameters such as CSS calculated?
No. What we would do in multiple doses is look at a particular interval. So here, if we look at steady state, for now, you would set the steady state tau, and it would do the NCA calculations based on that time interval. But if I understand the question correctly, we do not have a CSS calculation in the program. I know that. Something that could be added fairly easily. So one of the things we'd like to have is feedback on version 1 to say, what do you want to see in 1.1 or 1.5?
Well, can we just add a little bit to the steady state parameters? For multiple administration, I believe it assumes that you're at steady state. Excuse me. So the standard parameters are calculated based on a steady state profile.
But I don't recall having CSS as an output. If it's there, I just don't remember it.
Oh, C average. Okay.
Yeah.
Yep. Yeah, we should be adding that.
Simple calculation. We can add it easily if it's desired. Apparently it is. Next question.
That appears to be the last of the written questions. I see among our attendees that, let's see, Jeff Dolan was showing a hand raise. Let's see if he's still there. Oh, we haven't. Oh, Haiying. Oh, Haiying has just sent an answer. We have CAV calculation. Haiying has sent us that.
Okay, yeah. So yeah, it's the same thing. CAV, C average, or CSS, C steady state. So we just don't call it CSS. We call it C average. Thank you. I should have made that connection.
Okay, and again, Jeff Dolan is showing a hand raised, so I don't know if he wants to ask a question or make a comment. I'm unmuting. And Jeff, you can speak to us now.
That's funny, Renee. On my screen, the hand is down. Sorry about that.
Oh, no, you're still raising it.
That is strange. Can you hear me?
Have a comment, Jeff?
Yes.
You can hear me.
Yes, we can hear you. Do you have a comment?
Okay. No, I absolutely don't. I'm sorry. My hand is down on my screen. I don't know why it shows up on yours. Sorry about that.
Bye, Jeff.
Okay, well, we made the hour. I think we covered everybody's questions, and I'll turn it back over to Renee to wrap up.
Thank you all for this excellent and insightful discussion. So this concludes today's webinar. If you missed any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. Thank you, everyone, for joining us today, and have a wonderful rest of your day.